Hello everyone, welcome to the seminar today. Today we have Erik Amézquita with us, who will be talking about TDA to characterize spatial distributions, looking at mRNA at subcellular resolutions. Erik, take it away, the floor is yours.
Okay. Thank you, Bastian, Henry, and everybody else at AITRN for inviting me. My name is Erik. Currently, I'm a postdoc, soon to be faculty here at the University of Missouri, split between the Division of Plant Science and Technology and Math. So as you can infer from the title, my research and this talk will be focused on how to use tools from TDA, topological data analysis in an extremely applied plant biology setting. As any good interdisciplinary project, this cannot be possible unless you have a large team of different highly motivated people with plenty of different expertises. In particular, all the data and biological insights have been provided by Mark Liebold and his lab, also here at the University of Missouri. So before I jump straight into the contents of my research, just a bit about me. So my background is in mathematics. I did my bachelor's degree at Universidad de Guanajuato with extensive support from CIMAT, Mathematics Research Center, where I started applying TDA to non-mathematical disciplines. In this case, back in the day was archaeology. And I found those applications super fun. So I did my PhD in this new, at that time, well, still you can call it a new department, computational math, science, and engineering at Michigan State. To work with Liz Munch, and this department is super fun because it's all STEM departments at once. It's a hodgepodge of all disciplines, which in my case meant that I could take both math, statistics, and a lot of plant biology courses. And I like to joke that when I went to Michigan State, I went there for the math and I stayed for the plants. Then I moved to Missouri, University of Missouri, as a preparing future faculty postdoctoral fellow, split 80% between plants and 20% math. And starting September, I just signed the contract 2 days ago, so I'm still kind of internalizing the news that I will become an assistant professor here at MU. This time, at least for the time being, 100% in plants, but I'll see if I can get a slight appointment in math. So like I said, my overall research is about how to use techniques from TDA to plant biology data. In biology, they call this phenotyping, where a phenotype is a feature that is visually obvious, that is observable. So phenotypes can be patterns or movements or even data itself. So I know that I'm preaching to the choir here when it comes to TDA applications, that the way I see it is that TDA gives me this nice set of tools that allow me to quantify intuition, that visually you can tell that patterns here are interesting. So then we can use, say, persistence images, and this will be the focus of the talk, to quantify the intuition that we have when looking at different plant cells and the different patterns that mRNAs, different gene particles, make within the cell. Or we can be looking at movement. I know that usually you don't think plants and movement on the same sentence, but there are plants that do wiggle, and they wiggle a lot. What you are seeing here is Cuscuta, is a parasitic plant that wiggles and wraps around hosts, and once it wraps around the host, it will just suck the life out of the poor tomato plant. And the different environmental conditions will produce different wiggles, and then we can use, for example, say, persistent landscapes to quantify those wiggles, or we can have gene expression data, and we can use Mapper to cluster different samples or different cell types and see a continuum of how from one cell type we go another one. So for today, I will be focused mainly on this first part, but I'm more than happy to answer questions at the end about these other two projects and more things that are ongoing. So the roadmap for today, it will be a bit unorthodox for a math talk, where instead of giving you the math first, I will give you the biology first, and then I will jump straight into how we process that specific biological data and some results. So a crash course in genomic analysis, in case you are not familiar with biology. Biology, one of the most important concepts is what they call the central dogma. It sounds very profound, and in a way it is. But in very simple terms, you know that there is a cell. Inside the cell, there's a nucleus. Inside the nucleus, there is a copy of the whole genome of the cell. There's DNA, and then if you think about it, every single cell in your body or every single cell in your favorite plant will have the exact same DNA. However, an organism is composed out of very different cells, very different cell types. In our case, in humans, a neuron is not the same thing as a bone cell, and a bone cell is very different than a blood cell, and a blood cell is very different than a muscle cell. And still, all of these cells genetically are the same. So the difference is that the DNA has all the genes, but depending on the cell type, only a small subset of those genes will be transcribed into RNA and then translated into proteins. So you can think this RNA as as a copy of a gene particle, and that gene particle will become a protein, and it's this protein that tells the cell how to behave. Neurons will use a subset of neurons are neurons because they translate a very specific subset of proteins. Liver cells are liver cells because they encode a very specific subset of proteins. Bone cells are bone cells because they translate a very specific subset of proteins out of the DNA. And normally the way the genomic analysis works is that now we have the technology that either you are looking at a whole tissue or even at individual cells, and we can tell which of all the possible genes that can be transcribed, which are the most abundant, which are overexpressed. And depending on which genes are overexpressed or underexpressed, we can tell, we can have a profile of different cell types. Like if a cell is leaf, if it's root, if it's stem, if it's flower. However, counting gene expression is not the full picture because localization also matters. So, and this is where the Liebold lab here at Missouri comes into play, because they have access to this super cool technology that, I'll be honest, I have absolutely no clue how it works. To me, it still sounds very much like science fiction. And what this technology is capable of doing, molecular cartography, is that they can look at individuals. They can take a tissue. In this case, they are looking at the soybean nodule. If you don't know what a nodule is, just think as a part of the root. So they can look at the small root tissue of the soybean, and they can look at individual cells, and not just look at individual cells, but they can look at individual mRNA, individual gene particles as they go from the nucleus to anywhere else within the cell. So every single point that you see here is an individual gene particle of a very specific gene. So with molecular cartography, they are able to track more than 100 genes, assign different color markers, and then be able to tell of all the particles that are floating within the cell, of all the mRNA particles, tell apart which particle is from which gene. And they can do this for thousands and thousands of cells and hundreds and hundreds of genes. And as you can imagine, that's a lot of data. And when you start visualizing it, you immediately see that there are patterns. You can, for example, see here that the green gene is very much surrounding the cell walls, and the red gene is kind of scattered uniformly across the cytosol and then highly concentrated around the nucleus. So my interest as a mathematician is essentially to quantify patterns. So how patterny is a pattern? More specifically, how to model these patterns made by mRNA. And then use this mathematical model to see if different, not just different expression levels, but different localization patterns define different cell types. And if you're thinking along those lines, you immediately realize that one of the main challenges is that whatever model you propose to quantify these patterns has to be robust enough to take into account the diversity of cell size, cell shape, cell orientation, and the possibility that different distributions from annular to uniform to anywhere in between. So that's why we are thinking, again, just giving some foreshadow. That's why TDA seems to be the tool that can give us this robust framework. So before jumping into TDA, so with this data, traditionally what you do is you look at density of transcripts. So now you have your whole cell. You count how many transcripts of one specific gene are floating around the cell, and you just divide that number of transcripts by the size of the cell. That gives you transcriptomic density. And just by doing that very simple calculation, we can already see some interesting patterns. That, for example, at the center of the nodule or this root tissue, the transcriptomic density is much lower than at the outer side of the nodule. And that's true for these two genes that we'll be focusing on, O5G and 17G. However, density is not enough, because, for example, you can have cells and the same cell, but two different genes with essentially the same density, the same number of transcriptomic, the same number of mRNA particles floating around, but you can visually tell that the distribution is different. Same number of green points versus blue points, but very, very different spatial distributions. So this is why transcriptomic density is not enough to make the most out of this extremely rich data set. So finally, this is where, so TDA is where is the way to go, at least for this project, where we start with an individual cell and an individual gene. We look at individual mRNA particles within the cell. That's why the title says at subcellular resolutions, because we are looking within the cell. Then with a KDE, we can make a heat map, and then by having this heat map, then I pose the question that if we understand the shape of the heat map, we can understand the underlying spatial distribution of these mRNA particles. And to quantify the shape of the heat map, we'll do sub-level set persistence, get a persistence diagram, and do persistence images. So just to so that was everything in a single slide. So just to go now step by step. I said, so we have one cell, one gene. We do a heat map. So now, and in this case, this heat map is in 2D. Although keep in mind that our data is 3D, but I'm visualizing it as 2D because that's easier to exemplify. If you take this heat map and if you rotate it 90 degrees, now you'll have your high density points will be large peaks. For example, like this corner that we can tell that there's a lot of activity going on over this corner, and places where I have pretty much nothing going on, they will look like very deep valleys. And sure enough, here at the center of the cell, there's nothing going on. And like I said, now my goal is to understand the shape of this heat map of this landscape. I'm using landscape a bit liberally here because I know that persistent landscape is a very specific thing, but when I say landscape, I refer to this object and not the persistent landscape that was originally proposed by Peter Bubenik. So we do a sub-level set filtration of this landscape, and if we are looking at this object as a bird, from the bird point of view, looking from up, from top, at the very beginning, we just have one big hole, and then as we keep moving up, we have an island, so we have a new blob, and then we keep moving up just a little bit more, and this island has merged with the mainland. So now we don't have an island anymore. We now have a peninsula, which means that a blob has merged or a blob has died. If we keep going up, and then our hole has been broken up into two pieces. So now we have a new hole born. Same, we keep going up. This hole has been split into two. A new hole is born. A new hole is born here, and a new hole is born here. But then, if we just keep going up and up and up, eventually holes will start to be filled up. They are going to be sealed, or they will die. So now we register the death of a hole, the death of another hole here, one more death, another death, and a final death. If we just keep going up and up and up, nothing will change. And I know that plenty of people already know this toy example, but just as a just keep in mind that what we are achieving with this sub-level set filtration is that we are keeping track of precisely of the peaks, or in the biological context, we are keeping track of the places where the transcripts are the most concentrated, and that character using TDA to characterize how the hotspots of transcripts within the cell that has a lot of biological meaning. So now that we have this persistence diagram that in a way it characterizes precisely how separate and how distinct are my highly concentration spots within the cell for very specific genes, we can start doing math. Well, we can start doing data analysis. But as you might know, the persistence diagrams are very strange objects that sure they are nice because they are stable with respect to noise. So if my original heat map changes a little bit, then my original diagram will change just a little bit. But the problem is that it's hard to do anything interesting with these persistence objects other than doing bottleneck distance to say, or bachelor size distance to say how different they are. There are no unique means, and they are hard to put into machine learning pipelines. So instead, we are going to transform the persistence diagram into a more machine learning amenable object, namely persistence images. And what that means is that we simply take our persistence diagram, we rotate it 45 degrees clockwise. So we get a lifetime diagram, or what I call a lifetime diagram, because honestly, I've never seen a standard name for this diagram, where the y-axis is simply death minus birth. And then we do a persistent surface, which is another heat map, another KDE, except that the heat of each point will be highly dependent on its persistence, on how up is on the y-axis. So for example, here, even though I have only a single point in the corner, it has a lot of heat because it persisted a lot, because presumably it corresponds to my hottest spot in my transcriptomic spatial distribution. and points that have low persistence, low lifetime, that presumably corresponds to not so hot and not so interesting hotspots. They will have a low weight. And now that we have this image, we can simply discretize it by a certain number of pixels, and all of these pixels will have a, just like any image, they will have an intensity value, a number, and it is with these numbers that I can finally start doing standard statistics and machine learning with my original data. And I won't go through the details, but just rest assured that this pipeline is mathematically very well justified, that the persistence images are also stable, meaning that if your original distribution of transcripts changes a little bit, your heat map will only change a little bit, which means that your persistence diagram will change only a little bit, which eventually means that your persistence image, the values of your persistence image will change just a little bit. And if you want more details on how the persistence images work, you can ask Henry here. Well, he's at a conference right now, but he's on the call. He's one of the leading people behind the mathematics of persistence images. So, as you might have guessed, developing my main interest is to take the mathematical tools that other much smarter people than I have already developed, and use those tools to get novel results in the biology context. So like I've been saying, we have individual cells, and we have individual genes. So for each cell and gene pair, we can do the pipeline that I have just described, get a heat map, get a diagram, get a persistence image, and then for both H1 and H2 components, get their individual persistence image, and that will give you a long list of topological descriptors corresponding to the spatial, corresponding hopefully to the underlying spatial distribution of these cell gene pairs. So that means a lot of numbers for a lot of cells, and let's instead of going. The topological pipeline is already quite crazy, so instead of getting even crazier with machine learning, let's just try something super simple, which is PCA, just plain old principal component analysis using both the H1 and H2 persistence images, and we get this nice point cloud that we can, and then we can take a few sample points from this point cloud and see if they and see how the actual raw data looks like. And sure enough, it seems that our whole topological pipeline is helping PCA to distinguish different spatial patterns, going from patterns that look kind of uniform to patterns that look more annular in the PC1, and going from patterns that look kind of homogeneous to patterns that are more heterogeneous in intensity. And maybe this visualization is a bit clearer. Again, PC1 seems to be going from uniform to annular, although it's not really annular. It's more about distinct hotspots from not distinct hotspots to very, very distinct hotspots, and from hotspots with equal heat or hotspots with equal intensity to hotspots with varying degrees of intensity. So yes, so PC1 just number of distinct hotspots, which then we can see that is highly correlated to the cell size and to the number of transcripts within the cell. And PC2 is more about the, like I said, the heterogeneity of hotspot intensity, and it's mostly correlated to transcriptomic density. And we can repeat the pipeline. We can repeat the topological pipeline, get more persistence images, and get more PCAs, but this time looking at different genes. The same observations, the same interpretations of PC1 and PC2 seem to hold. PC1, number of different hotspots, and PC2 is the heterogeneity of intensities across the hotspots, which in a way is not surprising because that's the whole point of choosing sub-level set filtration, like I mentioned, that is keeping track of the location and intensity, not necessarily location, but separation between hotspots and their intensities. This is another gene, same interpretation of PC values. One more gene, just for illustration purposes, that again, just one hotspot, many different hotspots. All the hotspots seem to have the same heat. Some hotspots are hotter than others. Just one more. So does this mean anything biologically speaking? And the answer is yes. So like I mentioned at the beginning, we can go back and try to correlate now our PC values, that our summary of topological values to, again, eccentricity, or how far from the center the cell is with respect to the nodule, with respect to the root. And while PC1 doesn't seem has a very weak correlation, we have slightly better results when looking at PC2 or when looking at the heterogeneity of hotspots. Cells at the center have different values than cells at the outside. Or we can look at another gene, 17G, same results. PC1 doesn't tell us much, but PC2 seems to have, in this case, with this gene, an even better correlation between the topological descriptors at the center versus the topological descriptors of cells on the outside. And this is biologically interesting because we know for a fact that cells at the center are senescent, meaning that they are old and they are getting ready to die, because in a way, the nodules grow from the inside out. So the cells at the center, they are older, and the cells at the outside, they are much younger. So when it comes to cell senescence, or how the cell gets ready to pass away, what our results are telling us is that it's not just about the number of transcripts, but it's also how these transcripts are spatially located within the cell. So okay, so I've been just saying that PC1 and PC2 is just, in a way, another an alternative to quantify transcript number and transcript density, transcriptomic number and transcriptomic density. But can we do something else? And I say yes, because going back to our PCA, we can think this point cloud as our morphospace of all the observed mRNA spatial localizations. And when I look at these point clouds, one of the first things that I think about is, these corners, for instance, are completely empty. Why is that? Because in principle, I can take a point here, and I can work backward, and that will produce a persistence image that, in a way, I can associate that persistence image to a spatial distribution, which means that there are spatial distributions that, in theory, are possible. Well, mathematically are possible, but biologically they are impossible. Or if you want to be a bit more careful, that biologically are not observed in our data set. So there are distributions that even though mathematically are possible, there must be some biological constraints that do not allow that a spatial distribution of mRNAs to materialize. So to give you an example, in this case, I'm just taking one cell, and I thought of some very artificial distribution, which is just take 20 points within the cell equally spaced, and have them have each of these points have the same intensity, and then just add some noise. So an extremely ordered, extremely artificial distribution, but it helps to illustrate my point that you have these artificial distributions. You can do the heat maps. You can then extract persistence images, throw it back to the same PCA, and let's see where they land. And yes, this extremely artificial distribution lands way outside my observed point cloud of mRNA localizations. Same with a bit more, if I add a bit more noise, I'm still very much in artificial land, still very much artificial land, very artificial land. And as I keep adding more noise, I'm slowly but surely getting into more realistic observation, into more realistic patterns. This is another example where, again, it's just 20 points equally spaced across this one cell, except that the point at the center has, the cluster at the center has much higher intensity than the clusters on the outside. And this time I make the same observations that these first 4 distributions are extremely artificial. Sure enough, actually these first 5, they are extremely artificial, and they are well outside my observed point cloud, my observed PCA. But as I keep adding more noise, things get a bit more realistic. Again, this also showcases that PC2 keeps track of how diverse is the intensity within hotspots, between hotspots. Naturally, this is, again, these are just patterns that I thought immediately, but you can think of more structured ways or a more systematic ways to explore different patterns and see where they land in your pattern space. So let's, very well on time. So in conclusion, biologically speaking, we have this topological pipeline that allows us to extract information of the spatial patterns of different cells, different genes, regardless of the size, the orientation, and the shape of the cell in question. And we know that these spatial patterns values, these topological values, they are different from cells at the center than cells at the center of my nodule versus cells on the periphery of the nodule, which indicates that that spatial distribution is important for cell senescence. We also have a morphospace to explore what is mathematically possible versus biologically possible. Mathematically speaking, we have this is an example of quantifying patterns using TDA. And in principle, this pipeline, we have focused on 2 genes with additional explorations of other 4. But like I said, with molecular cartography, we have access to thousands of cells and hundreds of genes. So now we have this framework ready to enrich this morphospace, because right now this morphospace is only keeping track of 2 genes, but in principle we can incorporate information of the 100 genes that we have at our disposal, and not just 900 cells at the nodule, but 3,000 cells in the whole tissue. Presumably that will give us a much richer morphospace and much richer way to explore, again, mathematically possibilities versus biological possibilities. All this was coded on Python using scikit-learn, NumPy, scikit-learn, matplotlib. The TDA part was done with Goody and Persim. All of these, the manuscript is finally coming along. Hopefully we'll have a preprint in the next couple of weeks, and the preprint will contain a link to the GitHub where all this code will be available. And I want to thank again all these interdisciplinary projects are not possible unless you have a large team of people with very different skill sets. I have no clue how to use a pipette. That's why I need a team to provide me with the data and their biological insight, specifically the Libolt lab and their collaborators. And I want to thank Henry, Bastian, and the rest of the IATRN team and you, the audience, for bearing with me for the past 35 minutes.
Thank you very much. Let's all unmute and give a round of applause to Erik. So do we have any questions in the chat? I don't think so at the moment. So while people are waiting for some chat, oh no, we have one. Adam, feel free to ask that yourself, but otherwise I can also read it. So maybe I'm just going to do that. So Adam is asking, possibly a naive question, do you know if or how the height filtration of the KDE relates to standard multi-parameter persistent homology techniques?
Yeah, I'm not entirely sure if I can fully answer your question because I know multi-parameter persistence on a extremely high level. So most of what I know of multi-parameter persistence is that instead of looking at just height, you're looking at height and something else or two different parameters. But as soon as you have two parameters or more, things on the mathematical realm and even worse on the computational realm, things get extremely complicated. So that's why I stick to regular persistence, looking at a single parameter. And like I mentioned, I like this this height filtration of my KDE because in that way, even though I'm doing just one parameter height, indirectly what I'm doing is filtering by density, which in turn informs me about where the transcripts are the most located, which biologically is what has meaning. Where are my gene particles concentrated in the cell? All of this is part of a growing body of literature on mRNA localization, where like I said, the idea is that you don't keep track of just which genes are highly expressed, but where they are. In a way, you can think of it as building a house. It's much easier to transport the materials of a house to the new development that is happening on the outskirts of the city, as opposed to build a house in the center of the city and then just move the house as a whole to these suburbs. So that's why by focusing on height, we focus on density and we focus on this mRNA localization. All of this is to say that we only stuck to a single, that the important part is to stick to a single parameter because that makes math and computation so much easier.
Nice. We have another question by Dave, I think.
Yes. Thank you. I enjoyed the biology discussion. So my question, it's similar to the previous one, but have you looked at or are you able to look at more than one gene expression simultaneously? So with this, so in
yes, actually,
In a given cell, like, yeah, that's what
Yep, here.
the picture, the real data picture you went past, the nodal cells,
this one?
that picture, yes. Suppose you have something like that with multiple, and you're able to observe multiple genes. I don't know if you're able to. Can you then say something? Would you be able to say something about the relative densities of the two genes? So that perhaps something is going on where they're both high, one is low. And it seems like you might be able to do that, separating them and put both sets of gene persistent images through your PCA machine.
Yes.
I don't know what you get, but that's, yeah.
Yes, that's why I was going to this figure, because that's exactly what we did for these two genes.
Okay.
So in the nodule, in the section of the nodule of the root that we were focusing on, we were looking at about 900 cells.
Mm-hmm.
And we started focusing on two different genes, the 17G and 05G. And we chose these two because visually they are for a large number of cells, they have roughly the same number of transcripts. So if one is highly expressed in one cell, the other gene in that same cell will also be highly expressed, and the other way around. But you can see that they almost look like they are repelling each other. They are like sort of complements. So we have 900 cells, we have two genes. So in total, we have, and for each of these, for each pair, cell gene pair, we extracted their persistence images. And by the end of the day, you have this huge matrix with 1,800 pairs. So 900 for one gene, 900 for the other, and all their topological descriptors. And for those 1,800 cell gene pairs, that's where this PCA is coming from. So actually, this PCA, for example, these cells, I think these five or four are one gene, and the rest are the other gene. So it's possible to combine information of the two at the same time, and not just say how one gene is spatially distributed, but how one gene is spatially distributed with respect to another one.
Can you draw biological conclusions from this?
Yes. So I don't have it here, but because the interpretation of PC1 and PC2 remains consistent, we also try to look at I forgot to include this figure of correlating the PC2 of one gene and the PC2 of the first gene and the PC2 of the second gene. And we also found a moderate correlation, which was not surprising, because if you only look at their transcriptomic density and their transcript number, they are very much correlated.
Good. One more question. How much of the variation is captured by the first two principal components?
So in this case, it's 50 and 21, so it's more than 70%.
Good. Okay. Good. Thank you.
Thank you for your questions.
Thanks a lot. Do we have additional questions? Yes, Daniel, please go ahead.
Hi, hola, Erik. So this is maybe more of a suggestion than a question, or maybe a follow-up to the previous question. It seems like this is a prime candidate for using a multi-species or chromatic persistent homology approach, where you consider more than one type of cell in the machinery of computing persistent homology. Have you looked into this, or what are your thoughts of whether it would be relevant to your problem?
So what's the term that you use at the beginning?
Well, the what?
What is the term that you use at the beginning?
Oh, sorry. So yeah. So also the question, maybe if you're familiar with this. So there's a version of persistent homology called chromatic. There are several versions of this. One of them is called chromatic, and another one is called multi-species. And the idea is to incorporate in the computations of precision homology the fact that you have different types of cells or different types of genes here in your case, and then you create this version of precision homology that actually is giving you this type of correlation, spatial correlation between the two.
Well, I'll be honest, this is the first time that I'm hearing those terms. So,
oh, okay. So great. I mean, that's an opportunity for you to look that up.
Chromatic persistence, you said?
Chromatic persistent homology or multi-species persistent homology. Yeah.
Do you have any particular?
The chromatic one has nothing to do with chromatic homotopy theory, which was confusing at first. But
do you have any particular preferences?
I should. Give me a minute now. I'll put it on the chat.
Okay, perfect. Yes, sounds like something I should look into it.
Thanks for the suggestion. Any other questions?
I'd have a question.
Sure, go ahead.
So, Erik, as I was going through, it seems, and I could be wrong, that cell orientation is important for consistent results. Is that right? So, for example, say we took one of these cells, and I mean, if this ever happens, say the cell rotated, would that be considered now to have a different distribution, or is it not dependent on cell orientation?
In principle, it should not be dependent on cell orientation, and that's why we use purely topological approaches rather than geometrical approaches. And in principle, the orientation should not matter because, I mean, if you are looking at this cartoon, if you rotate your heat map, this heat map, the times where you have birth and death of blobs and holes should remain the same, which in turn should, and if they remain the same, the rest of the pipeline should remain the same. I don't know if your question was going on that direction.
Yeah, it was. No, that makes sense. And that's where I guess my question was in this translation, right? Because you have a heat map originally, and then translating to this new heat map that's based on this persistent homology is to kind of fix that problem of orientation, right? Am I understanding that correctly?
Yes.
Okay, thank you.
Great. Any other questions for Erik? Daniel's posting references. This is great. Then I have one about the machine learning aspects. So, do you also foresee that there would be a way to like have a, let's say, differentiable classifier or like optimization scheme in there? Because so far, if I understand this correctly, it's the information is static in the sense that the fingerprints or the ways that you describe these things, they're kind of manually picked, right? So would this also be kind of learnable or optimizable?
Yeah, I guess this comes back to these two slides where these two artificial distributions are. I made these examples because that's the first thing that crossed my head, and because I was able to code it in 5 minutes. I'm not an expert on differential equations or dynamical systems, and but I know that there's definitely a way to encode different patterns and diffusions and diffusion patterns and diffusion rules to have a much smoother continuum of how different patterns change, and that should give you a better understanding on how the patterns vary and how they are reflected in your overall biological space of patterns. Well, but again, that's not my expertise, which is to say that I'm more than happy to to discuss with people who have this kind of expertise.
Gotcha. No, thank you very much. No, it's a good food for thought, I guess. So, any other questions for Erik incoming? If not, I would say we thank the speaker again, and then we move to the offline portion, the non-recorded portion of the talk. So thank you very much, Erik.